Hi everyone, this video is about different types of transport, some water movement things, and some GLUTs. So we're going to start with two types of transport, the passive and active. And so passive first. So the big thing about passive is we don't use ATP. So it's passive, and that means that it's not doing the work itself, right? So we're not using any active like energy, so no ATP. So the reason why things can move without ATP is because we're going down the concentration gradient. So things are going to move from higher concentration to lower concentration. So we're using our concentration gradient. Okay, so we've got two main categories of passive transport. We've got our facilitated diffusion and simple diffusion. So let's do simple first. So simple diffusion. So simple diffusion, so these are the small molecules that are uncharged. All of passive is going to be uncharged molecules, but simple diffusion are very small molecules that can they're small enough to just slip right through the membrane. They don't need any help at all. So, so no protein channel required. They can just slip through. So we're saying it slips through the membrane. So we're going to think small and nonpolar molecules for our simple diffusion. Nonpolar, small molecules. So they're small enough and they're not charged. They're not going to interact with those lipids in the membrane. They can just slip right through. And then facilitated diffusion is our other type of passive. Facilitated. So facilitated diffusion, this is going to use a protein channel. So these things are still not going to need any energy. They're still going on they're still going with the concentration gradient, but they're going to be maybe some like bigger molecules, and these will actually be charged. So I think I misspoke earlier and I said something about charged molecules. So the charged molecules cannot go with simple diffusion, but charged molecules can go with facilitated diffusion. We call charged molecules ions. So first of all, so facilitated diffusion, they still don't use ATP, but they need some sort of protein channel to get through the membrane. So uses protein channel. Okay, the reason why they need a protein channel is because these are either charged, so they're ions, or they're a little bigger molecules, so like our glucose molecules will need facilitated diffusion. Those are like our GLUT4s. So think ions. Let's do this in blue. Oh no. There we go. Okay, so ions and bigger molecules. So think like glucose. Okay, so this next one is not technically categorized under passive diffusion, but it fits. So osmosis is going to not need any ATP either, so that's why we're going to count it under here. So the biggest thing you need to know about osmosis is that osmosis is only talking about the movement of water and nothing else. So water, that's very important to know. So the water is going to move to even out our solute concentration, and we'll draw some of this out in a second. So let's write that down. So we move the water moves to even out solute concentration. Solutes are like the other molecules that are in the mixture, like salt or something, or whatever. Okay. And it's also very important to know for osmosis that water isn't just like moving through by itself the whole time. It has to use special pores called aquaporins. Okay, so let's do that first, actually. So let's go over our hypotonic, isotonic, and hypertonic solution. So, okay, so we've got, you know what, we're gonna draw this up here. So draw 3 little like beakers here. So we'll make this one hypotonic. Isotonic. And hypertonic. Okay, so we'll have like a little red blood cell here in our liquid. Okay, so in a hypotonic solution, we have more molecules, more solute inside the cell than we do outside the cell. So we've got more, we're gonna represent that just like with dots, right? So we've got, I don't know, a good number of dots inside the cell and just a small amount outside the cell. So water is going to want to move into the cell to balance out that concentration. When water moves in, we are increasing the capacity, the amount of stuff in the cell overall. So it's going to swell. And because we're talking about a red blood cells in our bodies do not have cell walls. So it's going to swell and possibly burst. Can you see this down here? Nope. Okay, well, right over here. Swell and possibly burst. Right? Okay. We'll get to that next. So hypertonic is the complete opposite. So with hypertonic, we've got only like a small amount of other molecules inside the cell, but we've got a high concentration outside the cell. So water wants to go and even out this concentration, so it's going to move outside of the cell. So that is going to decrease the amount of stuff in the cell, and it's going to cause the cell to shrink. So shrink. Okay. And then isotonic is very happy. This is what we want in our body. So we're going to have about the same concentration inside as we do outside. So I'm just drawing the same amount of dots to illustrate that. So nothing happens. It's just happy, healthy, normal sized red blood cell. So I'll do this right here. Just, oops, my little way to remember this that has been helpful for me. So first let's look at this. So when I look at the words hypotonic and hypertonic, I look at this right here, this E and this O. So hypotonic, so think like the letter O is big, right? It looks like a big swollen cell. So think hypotonic, the cell is swelling. When you look at an E, I'll draw a little bigger. When you look at an E, this part right here, right, is tiny. It's tiny and shrunken compared to an O. So think like hypertonic, the cell is shrinking, if that helps you. Some word association there. Good, so that's that. So that's all of our passive stuff. Hopefully you have it all written down. If not, pause the video and keep writing. I'm gonna erase because I don't have a very big board, and we're gonna go over active. let's see. So active transport. So we said passive transport did not need energy. Active transport is the opposite. It's active. We need something to run it and get it moving, right? So active transport does need some sort of energy import, some sort of energy source. That's the word I'm looking for. So requires energy overall. It's different for secondary and primary, so that's why I'm just saying energy. And we'll get into what type for which one. The reason why it requires energy is because we're moving things against their concentration gradient. So naturally, things are going to want to go along its concentration gradient, and we're forcing it against its will to where it doesn't want to go. So we need energy to do that. So we're going against the concentration gradient. Okay, so we've got two big types of transport here, of active transport. So we've got primary and secondary. So primary active transport, we'll just put primary AT because I have a small board. Okay, so primary active transport is going to use ATP directly. So ATP is going to directly bind or interact with these carriers, and we're going to use that energy directly to move things against its concentration gradient. So, and then secondary active transport. Okay, so let me back up. So when primary is using ATP and it's pumping things against its concentration gradient, it's building up more of a concentration gradient. So it's creating a concentration gradient, and the molecules are going to want to flow back to where it got pumped from. So secondary active transport uses that concentration gradient as its source of energy. So secondary active transport depends on primary to work. So we'll say it does not directly use ATP. So it does not directly use ATP, but it uses the concentration gradient created by primary active transport. Okay. Sweet. So those are two types of active transport. I am going to have to erase these because I want more space on the board to draw these out. So if you don't have it written down, pause and do that. Here we go. Okay. So we're going to have a little line here. So pretend that this is our membrane of a cell. Okay, so this side is the outside. I'm gonna write this smaller. And then this side is the inside. So the outside is technically going to be like the lumen, so the inside of our intestines, and then the inside is the inside of the actual cell, so the inside of the enterocyte. Okay. So we'll do primary active transport first. So we're gonna draw over here our this is our transport. So we'll do primary. So what's gonna happen? ATP is going to come and directly interact with this primary active transport. So inside the cell, we have just a little bit of sodium. I'm gonna do this with a different color. Outside the cell, we have quite a lot of sodium because of this gradient, because of this pump, sorry. Okay. And then inside the cell, we have a lot of potassium. Lots of potassium. Okay, and then outside the cell, we just have a little bit. So naturally, remember, things want to go down its concentration gradient. They want to go from high to low concentration. So naturally, there's a lot of sodium up here and not very much here. So naturally, sodium is going to want to go this way, and potassium is going to want to go this way. But primary active transport says, too bad, I'm going to keep building up a stronger concentration gradient and moving you to where you don't want to go. So our primary active transport is going to take sodium and keep pumping it this way in the direction it does not want to go. And it's going to do the same for potassium and pump it this way. Okay, so you probably recognize this. This is your prime example. This is your sodium-potassium transport, which is very important to maintain cell size and solute concentration. So that's our primary active transport. Okay, something I skipped over, sorry. So sodium, when we're doing this movement, we're moving 2 sodium at a time out, sorry, 3 sodium at a time out, and we're moving 2 potassium in at a time. So I like to think like Na, I'm out of here, like Na is moving out, right? And then potassium is moving the opposite direction inside the cell. Right, so this is creating a gradient. We're pumping lots of sodium out and lots of potassium in. So we said secondary active transport takes advantage of the gradient created by primary. So over here, let's draw our secondary active transport. So this example, we're going to talk about a sodium glucose transporter, so SGLT1. Okay, so this is not directly going to use ATP. We're using this gradient. So let me just redraw a bunch of sodium over here to illustrate that we have that gradient. So lots of sodium outside and just a little bit inside. Right, and then glucose is the other thing that we're moving with secondary active transport, because we're moving 2 things. So glucose, we only have a little bit. I thought that was green, sorry. Oh, it is green, it's just really dark. Let's use a different green. Okay, so, nope, not that one. Sorry, folks. Okay, there we go, that's a good color. Glucose, we've got a little bit of glucose outside the cell, and we've got a lot of glucose inside the cell. Those are G's. So when we are trying to absorb sugar, we absorb a lot into the enterocyte, but then we get to a point where we have a higher concentration inside the cell than we do outside, but we still want to keep bringing it in because we still need to absorb glucose. So glucose, naturally, its gradient is going this way. It does not want to move inside the cell because this is the high concentration, so we have to force it inside the cell, right? In order to do that, we are allowing sodium to move along its concentration gradient where it does want to go, but we're only letting it do that if it takes a glucose along with it. Okay, so sodium, we're just letting it go happily down its concentration gradient, and then the toll, you know, so that it does, so to let it do that, it has to take a glucose along with it. Okay. So this is an example of a symporter. So we've got antiporters and symporters when we talk about secondary active transport. I'm gonna write this down here. So this is a symporter. So symport is where they're both moving in the same direction. So molecule 1 is going to always move down its concentration gradient, whether we're talking about symport or antiport. Molecule 2 is the one that differs. So for symport, molecule 2 is moving in the same direction as molecule 1, and then for antiport, molecule 2 would be moving in the opposite direction as sodium, or sorry, as molecule 1, which in this case is sodium. But glucose, for sodium glucose transport, it's always going to be symport. I would definitely know the difference between symport and antiport. Okay. Cool. So that's important to know. So we're going to erase this and give a big picture view of our GLUTs. Okay. So let's use this. So we're going to draw an overview of things first, and then we'll write in what we're talking about. So just draw along with me. So this is your lumen. Okay, we'll label it. These are your enterocytes. And then this is your bloodstream. Okay, so this is your lumen. So like the inside of your intestines, right? These are the enterocytes, so the cells in your intestines. So it's kind of squished together. That says enterocyte. And then this is the bloodstream. Good. Okay, so we eat some glazers or whatever you're into, right? So we've got a bunch of glucose in our lumen, and we need to absorb it. So in order to absorb it, we've got our sodium glucose transporter that is pretty much always going to be there, ready to work. So that's our sodium glucose, is GLT, sodium glucose transporter 1. I'm just gonna label 1 because this is not very much room. No, you know what, it's fine. I can label it. Okay, SGLT1. Okay, so those are sodium glucose transporters, and those are going to be moving glucose into the cell at a normal pace. And remember, those are types of secondary active transport. So at some point, we are getting enough glucose to go into the cell that sodium glucose transporter becomes saturated, and they need a little bit of help. So they get, so GLUT4 comes, and sorry, GLUT2, my bad. GLUT2 comes and inserts into the membrane. So we're just gonna draw a couple of those. So these are GLUT2 transporters, GLUT2, okay. So those are gonna help and move some glucose into the cell. Okay, so now we're getting glucose in here, so that's good. We're absorbing it, but it's still just in the cell, right? We need to like absorb it into our bloodstream so we can actually do something with it in other parts of the body. So GLUT2 is also going to be in this membrane. It was already here. It didn't need to be like called in like it did up here to save the day for sodium glucose transporter 1 that was getting saturated. So GLUT2 is just kind of like already hanging out here. Okay. So let's see. So then that's going to let some more glucose get into the bloodstream. So GLUT2 is the one that has a fairly high Km, right? So it's not going to be activated. That means it has a low affinity. So it's not going to be activated until we have a good amount of glucose concentration, a high amount of glucose concentration. So that's good because GLUT2 is the one that's going to go over to the pancreas and tell it that, okay, we're getting a lot of glucose into our bloodstream. We need to release insulin so that we can get it out of the bloodstream and get it into other parts of the body. Okay, so let's draw a little pancreas over here. What color do we want our pancreas to be? Let's do a pink pancreas, okay? Pink pancreas. That's your pancreas. This is actually a pancreatic beta cell. I guess it's not just your pancreas. So, pancreas beta cell. Okay, so your GLUT2 is gonna come and insert into this membrane here. And we're gonna get glucose into the cell. And that's gonna alert our beta cell, oh crap, we've got a really high concentration of glucose now because GLUT2 has such a high Km, and that's gonna cause the pancreas to release insulin. So now we release insulin. So, insulin is going to activate our GLUT4 molecules. So, we're going to draw, this is supposed to be muscle. So that's muscle. And then right here is going to be some fat, so an adipocyte cell. So we've got skeletal and cardiac muscle. And then adipose tissue. So the insulin causes GLUT4 to be activated, and GLUT4 is going to get glucose from the bloodstream into the adipocyte and into the skeletal and cardiac muscle cells. Right, so now we're going to get glucose going into there from the bloodstream. So we can actually use the sugar for what we need sugar for. So also, since GLUT2 isn't going to be activated to go to the pancreas beta cell until we have a lot of glucose, we have a high concentration of glucose, once we do release insulin, insulin is going to come up here and tell these extra GLUT2s that they can chill out and stop working because they're letting in a lot of extra glucose, and we have enough in our bloodstream already by this point. We don't need to keep getting it from the lumen. So insulin is also going to come and it's going to cause these to come out of the membrane because we don't need them right now anymore. Okay, so one of the big takeaways from this is that GLUT2 will insert on its own, but insulin pulls it out, and then GLUT4 requires insulin to insert and start working in the first place. Obviously, the rest of this is important too, but those are some pretty big distinctions to make there. and that GLUT2 is the one that alerts the beta cell to release insulin. So I think that is it for this section, you guys. Thank you for watching, and as always, reach out to me if you have any questions. Bye.